Hello, my name is Lydia. Today I will be discussing my IIS, which is titled Experimental Evolution in Daphnia magna, an investigation into the effects of ammonium nitrate exposure. I'm going to begin with a brief introduction of the topic. Pollution in general is a major environmental concern. Agriculture is one industry that releases a significant amount of pollution into our waterways. The three major chemicals that are used in agriculture are pesticides, herbicides, and fertilizers. Effects of pesticides have been researched significantly. In fact, researchers have found both changes in genetic expression and composition in response to pesticide exposure. The impact of fertilizers, on the other hand, has been researched significantly less. Ammonia, a nitrogenous chemical, has been studied a bit and has been seen to lead to oxidative stress in organisms exposed. Nitrate, another nitrogenous fertilizer, affects aquatic organisms' ability to carry oxygen. Ammonium nitrate is a fertilizer that has components of both the previous compounds and has been studied less, and there are some controversies regarding the effects. Some studies have found significant detriment to the health of organisms, while other studies have found no effect. With my study, I hope to fill in the gap in the research. Daphnia magna were chosen as the study species in this project due to ease of care, use as a model organism, full genome sequencing, the fact that they are a keystone species, and the fact that they have visual indicators of stress. Being a keystone species implies that the health of the ecosystem depends on this species. Because Daphnia provides a direct link between primary producers and other species, they are a keystone species in many freshwater environments. Therefore, if Daphnia are affected by a chemical, it could lead to changes throughout the whole ecosystem. Daphnia also have a few visual indicators. One, they express hemoglobin under stressful conditions, which shows as like a pink color. And two, they can change their mode of reproduction under stressful conditions. This provides an easy way to determine if the Daphnia are experiencing stress. One major goal of my study was to determine if there were lasting genetic effects on the populations of Daphnia magna exposed to ammonium nitrate. I did this through both microsatellite analysis and through analysis of genes of interest. Microsatellites are repeated regions in the genome that are a good genetic marker because they can change very quickly. By comparing microsatellite information in parental population to microsatellite information in final populations, genetic changes over time can be seen. I also investigated two genes of interest, heat shock protein 90, or HSP90. It's a protein that aids with protecting cells during oxidative stress, which has been previously seen in exposure to ammonia, which is one component of ammonium nitrate. HB4, hemoglobin subunit 4, is a subunit of hemoglobin. Because nitrate has been shown to make it so hemoglobin can no longer bind to oxygen, I thought it would be an interesting gene to study to see if there were any potential genetic impacts on hemoglobin. In general, my study had three major hypotheses. One, there will be changes in population dynamic metrics between control groups and treatment groups. Two, there will be a change in allele frequency between control and treatment groups. Three, there will be changes in genes of interest between the parental population and the final population of treatment groups. Additionally, I visually investigated hemoglobin expression in all groups to see if there was a difference between the control group and the treatment groups. Next, I'll be moving on to a brief overview of the methods used in this project. Generally, my research was an experimental evolution study, which means that I was investigating evolutionary questions in a laboratory setting. I essentially set up conditions for Daphnia to evolve so I could study if or how that evolution took place. So, in general, my experimental design is as follows. The Daphnia were split into three groups. One, a control group. Two, a group exposed to ecologically relevant concentrations of ammonium nitrate, and three, a group exposed to higher than ecologically relevant concentrations of ammonium nitrate. They were all kept in 1 liter containers and were allowed to evolve for 4 generations, or approximately 2 months. Daily, the number of individuals were recorded, along with number of individuals expressing hemoglobin. Prior to the start of the experiment, 15 individuals from each group were collected and DNA was extracted. This was repeated at the end of the experiment. PCR was run on all the samples to amplify both microsatellite regions and genes of interest, and all amplicons were sent to DNA analysis facility on Science Hill at Yale University for analysis. So what did I actually find? The population dynamics had some of the most interesting results in my study. In terms of growth rate, birth rate, and reproductive rate, the low concentration group performed the best. They had some of the highest birth rates and growth rates that indicated population growth, which was not seen in any other population. The high concentration group did have a population spike. However, in the next generation, the population quickly crashed. I hypothesized that the population could have been experiencing a trend called hormesis. This is when at low concentrations, the toxin has a stimulatory effect, while at high concentration, it has inhibitory effects. It is possible that ammonium nitrate could be a hormetic chemical to Daphnia magna. However, more research at different concentrations would be necessary in order to verify this. Interestingly, in microsatellite analysis, I was able to find some moderate genetic differences between population 2, which was the low concentration group. The differences were seen between both the control group and the high concentration group. Therefore, the differences seen in birth rates, death rates, and growth rates could have been attributed to slight genetic differences in the population from the beginning. Although these slight changes were seen, there were no significant changes throughout all the population, indicating no evidence of evolution in the population. Hemoglobin expression was observed throughout the entirety of the experiment. Throughout this observation, cyclical expression of hemoglobin was able to be seen, which is correlated with a new generation of Daphnia, as seen in the charts above. I hypothesized that this may be the case due to a specific signaling pathway that is present in Daphnia magna. Hemoglobin expression can occur in two ways, one through low oxygen, and two through the terpenoid signaling pathway. This pathway is also responsible for male sex determination, and it is possible that laboratory conditions could have led to slight stress levels that led to increased terpenoid signaling during reproductive times, also leading to increased hemoglobin expression towards the beginning of a new generation. HB4, a subunit of hemoglobin, was sequenced for all individuals in all treatment groups. However, no changes among the populations were able to be seen. This makes sense because the hemoglobin expression was consistent among all populations. Another gene I investigated was HSP90. As this gene helps with the stress response, I hypothesized that there may be a change in the gene leading to a better response. Interestingly, 3 different sequences were seen throughout. Through visual investigation, they appeared to occur equally in all populations. The first outcome as seen in this image is that the amino acid sequence aligned with the reference sequence, indicating no alterations. Outcome 2 is a nucleotide insertion at base pair 37. This led to a frame shift mutation that led to the introduction of many stop codons in the sequence. Most likely, this would make the version of the protein non-functional. A similar result was seen in outcome 3. A nucleotide insertion was present at both base pair 5 and at base pair 37. This also led to many stop codons and most likely a non-functional protein. It has been determined in previous research that HSP90 must be functional, or else it will lead to the death of the individual. Therefore, we hypothesize that a gene duplication event may have taken place in these populations. This would mean that somewhere else in the genome, a functional copy of this gene exists. This was unable to be verified with the methods that were completed in this research. However, because of the prominence of outcome 2 and 3, these mutations were most likely present in the starting population. Overall, I think there are 2 major takeaways from my project. One is that no evidence of evolution was found in Daphnia magna exposed to ammonium nitrate for 4 generations. And secondly, ammonium nitrate may be a hormetic toxin to Daphnia magna. This could lead to some interesting ecological impacts if this were to be the case. At low concentration of toxin, there would be a population boom, while at high concentrations, there could be a population crash. Both changes would impact the ecosystem as a whole. Overall, I feel that my research serves as a great starting point for further investigations into the impacts of fertilizers on Daphnia magna and therefore potential effects on the ecosystem. Thank you so much for listening. If you have any additional questions, you can feel free to stop by from 4 to 6, where I'll be here to answer questions live. Additionally, feel free to reach out at my email, lbruno21@worcester.edu. Thanks again and have a great day.